Search for fasting advice, and you will inevitably find a specific timeline. The claim is that at exactly 16 or 24 hours of fasting, a biological switch flips and your cells begin a deep cleaning process. This data comparison shows hepatic glycogen depletion rates, exposing the origin of that claim. On the left is a mouse, which has a metabolic rate roughly 7 times higher than a human. A 24-hour fast for a rodent completely depletes these energy stores, triggering a severe starvation event. On the right, the human curve shows a much slower, extended depletion. Projecting a rodent starvation timeline onto human biology is scientifically invalid. To determine if a predictable cellular reset actually occurs, we must audit direct human clinical data. Under nutrient abundance, the body runs an anabolic growth program. Insulin binds to cellular membrane receptors, triggering a rapid chemical cascade deep into the cell. This signaling cascade activates a protein complex known as mTOR C1. Acting as a strict molecular gatekeeper, it suppresses the cell's recycling machinery and forces the system to prioritize building and storing new material. During human fasting, a metabolic shift occurs. As insulin drops, mTOR-1 powers down, while the energy-sensing AMPK complex powers up. AMPK sounds the alarm to initiate recycling, but triggering this alert doesn't guarantee the cellular cleanup crew completes the job. This diagram shows an autophagy film sealing around cellular debris. Fasting guides cite the mass accumulation of these garbage bags, measured by LC32, as proof of cleaning. But this 72-hour human skeletal muscle graph reveals a flaw. LC32 and the debris-carrying protein P62 actually trend upward together. During successful autophagic degradation, P62 is destroyed along with the cellular waste, meaning its levels should drop. The simultaneous rise of both markers indicates a backlog in the waste degradation process, causing a bottleneck rather than a completed cellular cleanse. Inside the cell, the starvation response is tightly localized. The mitochondrial coordinator, SIRT3, aggressively accelerates fatty acid oxidation and ketone production to sustain energy levels. This metabolic response varies drastically by organ. The liver rapidly initiates recycling pathways to generate fuel for the brain, while human skeletal muscle actively resists breaking down its own proteins during short-term fasting. The human body compartmentalizes this stress to prioritize systemic survival. A synchronized full-body aesthetic reset triggered by a specific hour on the clock is a biological fiction. Treating human autophagy as an on-demand janitorial service that clocks in at exactly 16 hours ignores the extreme complexity and specific survival mechanisms of human biology. The data tells us to abandon rodent-based timelines. The goal of human fasting is better realized by focusing on its proven metabolic benefits, like improved insulin sensitivity and greater metabolic flexibility.